So here we are at Chapter 3, Topic 3.2, Prokaryotic External Cell Structure. For this topic, we have a lot of objectives. So let's go through. Describe the composition, function, and relevance to human health of glycocalyxes. Distinguish capsules from slime layers. Discuss the structure and function of bacterial flagella. Eventually, you should be able to differentiate between the structure and movement of bacterial and eukaryotic flagella. List and describe 4 bacterial flagella arrangements. Compare and contrast the structures and functions of fimbriae, pili, and flagella. Describe common shapes and arrangements of bacterial cells. Describe the sugar and peptide portions of peptidoglycan. Compare and contrast the cell walls of gram-positive and gram-negative bacteria in terms of structure and gram staining. Compare and contrast the cell walls of acid-fast bacteria with typical gram-positive cell walls. Diagram a phospholipid bilayer and explain its significance in reference to a cytoplasmic membrane. Explain the fluid mosaic model of membrane structure. Discuss how the structure of the cytoplasmic membrane aids in its cellular function. Describe the functions of a cytoplasmic membrane as they relate to permeability. Compare and contrast the passive and active processes by which materials cross a cytoplasmic membrane. Define osmosis, and distinguish among isotonic, hypertonic, and hypotonic solutions. Be able to describe the direction of water flow across the cytoplasmic membrane when given what type of solution is on each side of the membrane. Compare the structure and function of archaeal and bacterial fimbriae. Describe the structure and function of hammy. Contrast the types of archaeal cell walls with each other and with bacterial cell walls, and contrast the archaeal cytoplasmic membrane with that of bacteria. Whew, that's a lot, isn't it? But we will be covering all of these topics. Now, on to what structures we'll be covering in this topic. We will be discussing glycocalyces, flagella, fimbriae and pili, hammy, cell shapes and arrangements, cell walls, cytoplasmic membranes, a review of osmosis, and transport. In the next topic, we will be discussing internal structures. We will be discussing the structures of both bacteria and archaea, since they are both prokaryotic or lack a nucleus. We don't know as much about the archaea, since they have only recently been separated from bacteria. Under a light microscope, they look the same, so it's understandable. Later on, in Chapter 11, we will be discussing archaea and bacteria in more detail, but for now, make sure you know the differences and similarities between them. Some prokaryotic cells, both bacteria and archaea, have a sticky substance on the outside of their cells that we call a glycocalyx, which in Latin means sugar cup. We use that Latin term because usually the main component of the glycocalyx is, you guessed it, carbohydrates. They come in 2 flavors, capsules and slime layers. To quote from the textbook, when the glycocalyx of a bacterium is composed of repeating units of organic chemicals firmly attached to the cell surface, the glycocalyx is called a capsule. A loose, water-soluble glycocalyx is called a slime layer. Both a capsule and a slime layer help protect the cell from drying out. Out in the wild, unpredictable world, that's an important function. They can help prevent a cell from being phagocytosed or swallowed down by a predatory eukaryotic cell without a cell wall, such as an amoeba or a white blood cell. Usually, a capsule performs that function. Both capsules and slime layers can help the cell attach to surfaces, such as your teeth or sand granules in a stream, or to other cells. Next up, flagella. Flagella are long, proteinous extensions that enable the cell to move through the environment. The protein in bacteria flagella is called flagellin. Very convenient for remembering, isn't it? The flagella of archaea are not composed of flagellin, but they have the same overall structure as shown in these diagrams. All prokaryotic flagella are composed of 3 parts. The filament, here it is in this diagram, which is a long thread-like structure that sticks out from the cell wall. And here, the rod anchors the filament into the cell wall. Then there are the protein rings with ball-bearing like proteins between them. All this allows the filament, the flagellum, to move 360 degrees like little outboard motors. Prokaryotes, both archaea and bacteria, may have more than one flagellum, as we'll discuss in more detail in the next slide. In bacteria, if the flagellum move counterclockwise, they tangle together and act as one flagellum to propel the cell in a straight line, which we call a run. When they move clockwise, the flagella separate and buzz independently. This causes the bacterium to move in a random fashion called a tumble. Runs happen more often when the cell is moving towards something it likes, be it a food source or light in photosynthetic bacteria. When a cell moves toward a chemical, such as sugar, didn't think sugar was a chemical, huh? We call that positive chemotaxis. Movement toward light would be, that's right, positive phototaxis. Tumbles, on the other hand, occur more frequently when the cell is moving away from something. Many bacteria are killed by light, so they would exhibit negative phototaxis by moving away from the light. Got it? Archaea flagella tangle together no matter the direction they spin. Also, archaeal flagella are smaller, grow in a different fashion, and often have sugar molecules that attach to the proteins. I'll leave you to make a list of the other differences from the textbook. Let's talk flagellar placement. Why, you may ask, are we learning all these terms? Because prokaryotes don't have a lot of things to distinguish between the different species and genuses, so we latch on to what we can, such as placement of flagella, as a part of characterizing them. First off, we use the Latin term for hair, trichus, for a lot of these terms. So this bacterium would be monotrichus, mono for one and trichus for hair. So this bacterium has one hair or flagellum. Because it's rod-shaped, or a bacillus, we can add polar to the description as in polar monotrichus, which means it's coming out of one end rather than the middle. This bacterium has a tuft of polar flagella. This bacterium has a flagellum coming out of each end. We used to call that bipolar, but we've changed the name to Lophotrichus. I can't imagine why. And this puppy, with flagella all over the place, is peritrichus. There are other extensions from the cell surface other than flagella. Fimbriae and pili are both generally shorter than flagella and they don't spin. Fimbriae are rods that the cell uses to attach to both surfaces, other prokaryotic cells, and eukaryotic cells. They can be used in cell movement. Once the fimbriae attaches, the cell can shorten it, towing the cell toward what it is attached to. Pili are hollow rods that are sometimes used to transfer genetic material between cells, like what's happening in this micrograph. They don't transfer the entire genome, so it isn't sexual reproduction. It's more like sharing notes before an exam. We'll talk about pili and horizontal transfer of genetic material in a later chapter. Archaea have fimbriae, like shown here. They also have a cool structure called hami, or hammy, I'm not sure how it's pronounced. It's like a grappling hook on barbed wire. And, like all grappling hooks, they are used to grab a hold of stuff, like surfaces and other cells. Cells come in two basic shapes, round and rod-shaped. Well, there are other shapes, but they are rare. Rods can lengthen and become comma-shaped like in this picture, or helical like in this picture. The circles are called cocci, the rods are called bacilli, and the commas are called vibrio. Helical bacteria have two terms, spirillum and spirochetes. Spirillum are rigid and often have flagella coming out of each end, so you know this one's a spirillum. Spirochetes are flexible and have flagella under a sheath. The flagella, which are kind of like ingrown hairs, are called endoflagella. When the endoflagella rotate, they spin the entire cell like, well, a corkscrew, and they bore their way forward. Archaea can come in all sorts of funky shapes, like squares and stars, like in this diagram, or sea slug shaped. I made up that last one. They have some lovely micrographs in your textbook, so go ahead and take a look at those. They can come in the standard cocci and bacillus shapes too. For arrangements, we'll be getting into that in more detail in Chapter 11, but let's go over some terms real quick in this micrograph that has several different sizes, shapes, and cell arrangements. If a cell is by itself, we call that a single cocci or bacillus. So here's a bacillus, there's a coccus. If they come in strings, looks like we've only got some bacillus strings, we call those streptobacilli, or streptococci of strep fame. Now, if they form clusters, these aren't real good clusters because they're not attached. But if they were attached and they're cocci, we would call them staphylococci. Staphylo is Greek for grape-like clusters, so when you get round bacteria looking like grape-like clusters, we call them staphylococci. Bacilli cannot form staphylo-type clusters. We'll go into why in Chapter 11, but for now, remember there's no such thing as staphylobacilli. Hint, hint.